Module 5, Objective 3, describe the locations and functions of the various types of neuroglia. Neuroglia support and protect neurons, making up about half of the volume of the nervous system, and there's various types of neuroglia found within the central nervous system and the peripheral nervous system. If you recall, in a previous lecture, we introduced the two main cell types of the nervous system. We had the neurons, which are responsible for carrying electrical impulses or action potentials through the body. And then we have these glia cells, which are supportive in nature. They support and help protect the neurons. Types of neuroglia in the central nervous system include 4. We have the astrocytes, the ependymal cells, the oligodendrocytes, and the microglia. We'll go over each one in turn. Astrocytes have very large cell bodies with a lot of processes. They tend to terminate in these little footpad-like structures that are called podocytes, and they function to maintain the blood-brain barrier. In fact, we can see how this astrocyte in this image is completely covering this blood vessel right here. And in doing so, it creates a barrier between the brain and the blood. In addition to maintaining the blood-brain barrier, astrocytes create a network, a three-dimensional network for the central nervous system. They help repair any type of damage found within the nervous tissue. They guide neuron development as well as control the interstitial environment. Ependymal cells form an epithelium that lines the central canals of the spinal cord and the ventricles of the brain. In lab, we'll talk more about the ventricles of the brain, but these are large bodies of fluid, large pockets of fluid within the brain. And the ventricles of the brain are actually continuous with this central canal of the spinal cord. And all of these structures are lined with these ependymal cells. Ependymal cells produce cerebral spinal fluid, which helps to circulate throughout the internal neural tissue. They also contain cilia that help circulate CSF. Oligodendrocytes, they have very, very small cell bodies with very few processes, and they create the myelin sheath. Myelin sheaths, myelin itself is a fatty substance that appears white in gross dissection. And myelin will actually wrap around axons. And when you have a myelin sheath on an axon, it's referred to as a myelinated axon. And the main function of myelinated axons is to increase the speeds of action potentials. When we look at oligodendrocytes, we have some terminology that we can use, such as internodes and nodes. Internodes are segments of myelinated axons, while nodes are nodes of Ranvier, lie between the internodes. And we'll show an image in just a minute of these two structures. In fact, we'll show them right now. Here we have the internode, so this region of myelinated axon. And you can see how this oligodendrocyte, here's your cell body, and it literally wraps around the axon, creating the myelinated axon. And the region that is wrapped is referred to as the internode. And what that leaves is the node, the little segment of unmyelinated axon that is exposed between cells. So here we have oligodendrocyte number 1, here's oligodendrocyte number 2, and between these two cells is a small node that is not myelinated. So internodes versus nodes. Microglia are the smallest and least numerous neuroglia within the central nervous system. They have many fine branched processes, and they simply migrate through the nervous tissue, and they phagocytize cellular debris and waste products and any type of pathogens. They're sort of the cleanup crew, if you will. Neuroglia of the PNS help to insulate neuronal cell bodies and most axons, and that we have two main types. We have the satellite cells and the Schwann cells. Satellite cells surround the ganglia. Remember, ganglia are clusters of cell bodies within the peripheral nervous system. So the satellite cells, they surround them, just as satellites surround the Earth. And they work in regulating the interstitial fluid around those neurons. The Schwann cells, they form myelin sheaths around axons of the PNS. So in a very similar fashion as we saw with the oligodendrocytes of the central nervous systems, Schwann cells myelinate axons within the peripheral nervous system. Here we see a typical neuron with multiple Schwann cells. In fact, each one of these little sections of internodes represent a Schwann cell. So here's Schwann cell 1, 2, 3, 4, so on and so forth. So it takes multiple cells to myelinate a single axon. Here we see that again as the Schwann cell is surrounding and ultimately myelinating these axons.